Hello there, and welcome to Science with Sleepy. I'm so glad you found your way here. Maybe you had a long day, or maybe you're the kind of person who likes falling asleep to deep thoughts and scientific mysteries rather than counting sheep. Either way, tonight's journey is for you. You've got nothing to do but rest. Let your mind relax, your breathing slow, and your body grow light, as if gravity itself has softened. Because tonight, we're going on a voyage, not through space, but through time. We're traveling back to the year 1831. Imagine you're a young scientist named Charles Darwin. You're standing on the deck of a ship called the HMS Beagle. The ocean stretches out before you in endless shades of blue and grey. Unknown lands wait ahead. Strange creatures you've never seen. Plants that don't grow anywhere else. Fossils that tell ancient stories. You're about to sail around the world for 5 years. 5 years of exploring, 5 years of collecting, 5 years of observing. You'll visit places no scientist has been. You'll see things that will change how we understand life itself. You'll notice patterns. You'll ask questions. You'll piece together a puzzle that no one has solved before. But here's the thing. You can't see the invisible code that connects all life. You can't read the secret instructions written in every cell. You can observe the finches with different beaks. You can see the tortoises with different shells. You can find the fossils that look like living animals. You can think, you can wonder, you can connect the dots, but the deepest truth remains hidden. The code, the mechanism, the actual instructions that make it all work. What if it didn't? What if Darwin had DNA sequencing technology? What if he could read the code of life itself? What if he could see the genetic differences, the mutations, the changes written in molecules? How would that have changed everything? How would that have changed science? How would that have changed the world? That's our journey tonight. We'll explore what Darwin really discovered with just his eyes and his mind. We'll learn how DNA sequencing works, how it reads the code of life, how it reveals the secrets hidden in every cell. We'll see how DNA has confirmed Darwin's ideas, how it proved he was right, how it showed the mechanism he could only imagine. And we'll imagine what might have happened if he had this incredible tool. What discoveries could he have made? What proof could he have shown? How would biology have advanced? How would medicine have changed? How would agriculture have transformed? The ripple effects would be enormous. One technology, in the right hands, at the right time, changing everything. Some of what you hear tonight may surprise you. Some may spark curiosity. Some may make you wonder. All of it is designed to help you drift gently into sleep, to let your mind explore while your body rests, to learn while you dream. And just before we begin, if these calm, curious explorations help you unwind or give you a quiet way to end your day, consider liking and subscribing to the channel. It helps others find their way here too, one sleepy soul at a time. Now let's begin our voyage, back through time, to a ship, to a scientist, to a discovery that changed the world. In 1831, Charles Darwin was 22 years old. He had just finished university at Cambridge. He wasn't sure what he wanted to do with his life. His father wanted him to become a doctor or a clergyman. But Darwin had other interests. He loved nature. He loved collecting. He loved observing the world around him. Then an opportunity came. A ship called the HMS Beagle needed a naturalist. Someone to study plants and animals during a long voyage around the world. The captain, Robert Fitzroy, wanted a gentleman companion. Someone educated. Someone curious. Someone who could help document the journey. Darwin's father didn't want him to go. He thought it was a waste of time, a dangerous adventure with no purpose. But Darwin's uncle convinced him. He saw the opportunity, the chance to explore, the chance to learn, the chance to see the world. And so, on December 27, 1831, Darwin set sail from Plymouth, England. The voyage would last 5 years. 5 years of exploring, 5 years of collecting, 5 years of observing. Darwin would visit places no scientist had been. He would see things that would change how we understand life itself. He would collect thousands of specimens. He would fill notebooks with observations. He would sketch what he saw. He would measure everything carefully. But he had no microscope powerful enough to see cells clearly. He had no way to read DNA. He had no understanding of genetics. He had only his eyes, his notebook, his pencil, his curious mind, and that would be enough. More than enough. Because observation is powerful. Because curiosity drives discovery. Because thinking can reveal truth, even without the best tools. The ship sailed from England on December 27, 1831. It crossed the Atlantic Ocean. The journey was rough. Darwin was seasick for much of the voyage. He spent days in his hammock, nauseous, uncomfortable. But he didn't let that stop him. When he could, he went on deck. He collected specimens whenever possible. He observed everything. He took notes constantly. The Beagle was a small ship, only 90 feet long. It carried 74 people. It was cramped. It was uncomfortable. But it was home for 5 years. Darwin had a small cabin. He shared it with the ship's artist. He stored his specimens there, his notebooks, his collections, his observations, all in that tiny space. The voyage took them to many places: Brazil, Argentina, Chile, the Galapagos, Tahiti, Australia, New Zealand, South Africa, many islands, many continents, many ecosystems. Darwin saw incredible diversity. He collected thousands of specimens. He filled notebook after notebook. He observed. He thought. He wondered. The ship visited South America. Darwin explored the continent for months. He traveled inland. He climbed mountains. He crossed rivers. He explored forests. He collected plants, hundreds of them. He collected animals, dozens of species. He found fossils, ancient bones, preserved in rock, telling stories of the past. In Argentina, he discovered the bones of giant sloths. These animals were enormous. Some weighed over a ton. Much bigger than the sloths that still lived there. Paleontologists now know these were Megatherium, giant ground sloths that stood over 20 feet tall when upright. They were extinct, gone forever, but their bones remained, preserved in rock, waiting to be found, waiting to tell their story. Darwin studied these fossils carefully. He measured them, he sketched them, he compared them to living sloths. The similarities were striking. The same basic structure, the same bones, the same shape. Just different sizes, different proportions, but clearly related, clearly connected. Why would that be? The question stayed with him. It haunted him. It drove him to think deeper. According to Darwin's own notes, he found these fossils near Bahia Blanca, in Argentina. He wrote about them in his journal. He described their size, their structure, their similarities to living sloths. These observations would later support his theory of evolution. They showed that species could change, that extinct animals could be related to living ones, that life had a history, a story, a connection. The ship went around Cape Horn, the southern tip of South America, one of the most dangerous passages in the world. The weather was terrible. Storms raged, high waves crashed, cold winds howled, the ship rocked violently. Darwin was seasick, constantly. But he didn't let that stop him. He kept observing, he kept collecting, he kept thinking, he kept asking questions. His curiosity was stronger than his discomfort. Then, in September 1835, the ship reached the Galapagos Islands. These islands would become famous because of what Darwin found there. But at the time, they were just a group of volcanic islands in the Pacific Ocean. Remote, isolated, about 600 miles off the coast of Ecuador. Full of strange creatures found nowhere else on Earth. The islands were formed by volcanoes. They rose from the ocean floor. They were never connected to the mainland. They're about 600 miles off the coast of Ecuador. Life had to reach them somehow. By flying, by floating, by drifting on the wind, by riding on logs, by hitching rides on birds. And once life arrived, it evolved in isolation. It changed, it adapted, it became unique. Each island is different, different sizes, different shapes, different elevations, different climates, different plants, different environments. This created different challenges, different opportunities, different pressures. Life on each island evolved differently. It adapted to its specific environment. It became unique. It became its own species. Darwin spent 5 weeks exploring the Galapagos. From September 15th to October 20th, 1835, he collected everything he could: birds, plants, insects, reptiles. He took detailed notes. He sketched what he saw. He preserved specimens to take home. He labeled everything carefully. Which island it came from, when he found it, what it looked like. He collected 13 species of finches. But he didn't realize their importance at first. He also collected mockingbirds. They were different on each island too. He collected iguanas. Marine iguanas that swim in the ocean. Land iguanas that live on the islands. Both unique. Both found nowhere else. He collected plants. Over 200 species, many found only on the Galapagos. All unique, all evolved in isolation. But he noticed something odd. The finches on one island looked different from finches on another island. Their beaks were different shapes. Some were thick and strong, like nutcrackers. Others were thin and pointed, like tweezers. Some were medium-sized, some were curved, some were straight. Why would that be? Darwin collected finches from different islands. He labeled them carefully. But he didn't label which island each came from. He thought they were different types of birds. He didn't realize they were all finches. It was only later, back in England, that an ornithologist told him they were all finches. Just different species, different varieties, but all finches. He didn't understand it at first. He didn't even realize they were all finches. He thought some might be different types of birds. But the question stayed with him. It nagged at him. Why would birds on different islands look different? They were so close together, just a few miles apart. Some islands were only 10 miles away from each other. Why would they be different? Modern scientists have studied these finches extensively. Biologists Peter and Rosemary Grant spent over 40 years studying them. They've identified 13 different species, each adapted to its island, each with a different beak, each with a different diet, each with a different lifestyle, all from a common ancestor, all evolved through natural selection, all proof of evolution, all discovered by Darwin's observation, all confirmed by modern genetics. The Grants watched evolution happen in real time. They measured beaks, they tracked populations, they saw natural selection in action. During droughts, finches with larger beaks survived better. They could crack the harder seeds that remained. Their genes spread. The population changed. Evolution happened right before their eyes, over just a few years. It was incredible. It was proof. It was evolution in action. Scientists have sequenced the DNA of all 13 finch species. They've compared the genetic codes. They've found the exact mutations, the specific changes in the code, the genes that control beak shape, the genes that determine size, the genes that create adaptations. They've seen evolution at the molecular level. They've confirmed Darwin's observations. They've proven natural selection works, all through DNA sequencing, all through genetic analysis. One gene, called ALX1, controls beak shape. Mutations in this gene create different beaks: thick beaks, thin beaks, curved beaks, straight beaks, all from changes in one gene, all from mutations in the code, all from evolution, all from natural selection. Scientists have studied this gene in detail. They've seen how it works, how it controls development, how mutations change the shape, how natural selection favors certain mutations. They've watched evolution happen. In the lab, in nature, in real time, they've seen the mechanism, the process, the truth. Another gene, called HMGA2, also affects beak size. Mutations in this gene make beaks larger or smaller. Combined with ALX1, these genes create all the variation, all the different beak shapes, all the adaptations. All from just a few genes, all from mutations, all from natural selection. Darwin saw the results, he saw the different beaks, he understood they were adaptations. But he couldn't see the mechanism. Modern genetics sees it. The code, the mutations, the genes, the process, the truth. It's all there, in the DNA. Written in code. Revealed by science. The Beagle returned to England in October 1836. Darwin had been gone for almost 5 years. He came back with thousands of specimens. Plants, animals, fossils, all carefully preserved, all carefully labeled, all waiting to be studied. He spent the next few years organizing his collections. He sent specimens to experts. Botanists studied his plants. Zoologists studied his animals. Geologists studied his fossils. He read their reports. He studied their findings. He thought about what he had seen. He wondered about the patterns. He questioned the connections. He wrote about his voyage. He published his journal. He described what he saw. He shared his observations. But he didn't publish his theory yet. Not yet. He needed more evidence. He needed to be sure. He needed to answer every question. He needed to build his case. So he kept working, he kept studying, he kept thinking. He spent years on barnacles. He spent years on pigeons. He spent years reading. He spent years corresponding. He spent years building his knowledge. He spent years preparing. He was patient. He was thorough. He was dedicated. Years later, back in England, he would think about those finches. He would study the specimens he collected. He would compare them. He would realize that each island had different food, different environments, different challenges. The question that had nagged him on the voyage finally had an answer. The patterns made sense. The connections were clear. On one island, the main food was hard seeds, large seeds with tough shells. Birds needed strong, thick beaks to crack them open. So the finches on that island had thick beaks. They could crack the seeds. They could eat. They could survive. On another island, the main food was insects. Small insects that lived in tiny spaces, in cracks in rocks, under tree bark. Birds needed thin, pointed beaks to reach them. So the finches on that island had thin beaks. They could reach the insects. They could eat. They could survive. On yet another island, the main food was cactus flowers. The finches there had beaks adapted for that. They could reach into the flowers. They could get the nectar. They could eat. They could survive. The birds had adapted. They had changed to fit their environments. Over time, the finches with beaks that worked best survived. They had more babies. Their babies inherited those beaks. The finches with beaks that didn't work as well died. They had fewer babies. Over many generations, the beaks changed. They evolved. They adapted. This was natural selection, the idea that would change science forever, the process by which life adapts, the mechanism of evolution. But Darwin figured this out by looking, by comparing, by thinking. He had no DNA evidence. He couldn't see the genetic changes. He couldn't read the code. He could only see the results, the different beaks, the different shapes, the adaptations. Imagine if he could have sequenced the DNA of those finches. Imagine if he could have seen the exact genetic differences, the changes in the code that made one beak thick and another thin, the mutations, the variations, the instructions written in A, T, C, and G. He would have had proof, immediate proof, not just observations, not just patterns, but the actual mechanism, the genetic code, the instructions, the proof written in molecules. He could have shown exactly how the beaks evolved, how the code changed, how mutations led to different shapes, how natural selection worked at the genetic level. But Darwin didn't have that. He didn't have DNA sequencing. He didn't even know DNA existed. So he did something remarkable. He spent 20 years gathering evidence, building his case, proving his theory, piece by piece, observation by observation, fact by fact. He studied barnacles for 8 years. Why barnacles? Because they showed variation, because they showed how species could change. He collected thousands of barnacle specimens. He studied them under a microscope. He compared them. He saw the differences. He saw the patterns. He understood how they evolved. He bred pigeons. Why pigeons? Because he could see selection in action. He could breed for specific traits. He could see how characteristics were passed down. He could understand inheritance. He could see how artificial selection worked. And if artificial selection worked, why wouldn't natural selection work too? He read everything he could find. Books about geology, books about biology, books about travel, books about other scientists, observations. He corresponded with scientists around the world. He asked questions. He shared ideas. He built his knowledge. Slowly, carefully, patiently. He built his case. Observation by observation, fact by fact, evidence by evidence. He didn't rush. He didn't publish until he was ready, until he had enough proof, until he could answer the critics, until he could explain everything. Finally, in 1859, he published his book, On the Origin of Species by Means of Natural Selection. It was revolutionary. It explained how life changes over time, how species adapt, how all life is connected, how evolution works, how natural selection drives change. The first printing sold out in one day. All 250 copies. Gone. Immediately. People wanted to read it. They wanted to understand it. They wanted to debate it. It was that important, that revolutionary, that controversial. A second printing came out quickly, then a third, then a fourth. The book kept selling. It kept being discussed. It kept being debated. By 1872, it was in its 6th edition. Darwin kept revising it. He kept adding evidence. He kept responding to critics. It became one of the most important books ever written. It changed science. It changed society. It changed how we see ourselves. Today, historians consider it one of the most influential books in history. It's been translated into over 30 languages. It's been read by millions. It's shaped how we understand life, how we understand ourselves, how we understand our place in nature, all from one scientist's observations, all from careful thinking, all from curiosity. The book was controversial. Many people didn't believe it. They couldn't see the mechanism. They couldn't understand how it worked. They thought species were fixed, unchanging, created separately. They couldn't accept that life evolved, that humans were related to other animals, that all life shared a common ancestor. Religious leaders criticized it. They said it contradicted the Bible. They said it was heresy. They said it was wrong. Scientists debated it. Some supported it. Some opposed it. The public discussed it. It was everywhere. In newspapers, in magazines, in conversations, in debates. Everyone was talking about it. Some loved it. Some hated it. But everyone had an opinion. It changed how people thought about life, about nature, about themselves, about their place in the world. Even today, over 160 years later, the book is still read, still discussed, still relevant. It's been translated into dozens of languages. It's been reprinted hundreds of times. It's one of the most influential books in history. All because Darwin observed. All because he thought. All because he discovered the truth. Darwin had evidence. Lots of evidence. Observations. Comparisons. Patterns. But he didn't have the deepest proof. He couldn't show the genetic mechanism. He couldn't read the code. He couldn't see the DNA changes. He could only show the results, the adaptations, the evolution, but not the mechanism itself. What if he had DNA sequencing? What if he could have shown the genetic code, the actual changes in the instructions, the mutations, the variations? The proof would have been undeniable. The mechanism would have been clear. Critics couldn't have argued. The code doesn't lie. The genetic differences would have been obvious. Evolution would have been proven, immediately, completely, undeniably. But he didn't have it, so he used what he had: his eyes, his mind, his patience, his dedication. And he changed the world anyway. He discovered one of science's greatest ideas. He explained how life works. He showed that evolution is real, all without seeing DNA. All without reading the code. All with just observation, just thinking, just curiosity. Darwin was a master observer. He noticed things others missed. He saw patterns. He connected dots. He asked questions. He thought deeply. That's what made him great. Not the tools, the thinking, the observation, the curiosity. On the Galapagos Islands, he found giant tortoises. These were enormous creatures. Some weighed over 500 pounds. They moved slowly. They lived for over 100 years. They were ancient. They were magnificent. And each island had its own kind, its own species, its own unique tortoise. The tortoises on islands with tall plants had long necks. They could stretch up. They could reach the leaves. They could eat. The tortoises on islands with low plants had short necks. They didn't need to stretch. The food was right there. They could eat. Their shells were different shapes too. Some were dome-shaped, round, smooth, like a helmet. Others were saddle-shaped, curved upward at the front, like a saddle. Why would that be? Darwin thought about it. He observed. He compared. He realized that the saddle-shaped shells allowed the tortoises to stretch their necks higher, to reach taller plants. The dome-shaped shells were for tortoises that didn't need to stretch, that ate low plants, that lived in different environments. Each island was different. The plants were different. The terrain was different. The climate was different. The tortoises had adapted. Over thousands of years, they had changed to fit their homes. They had evolved. They had become perfectly suited to their environments. Darwin saw this pattern. He connected the dots. He realized that life wasn't fixed. That species weren't created separately and never changed. Life was always changing, always adapting, always evolving. It was a process. A beautiful, ongoing process of change and adaptation. He also found fossils. In South America, he discovered the bones of giant sloths. These animals were enormous, much bigger than the sloths that still lived there. They were extinct, gone, but their bones remained, preserved in rock, telling a story. Darwin studied these fossils. He compared them to living sloths. He noticed the similarities, the same basic structure, the same bones, just different sizes, different shapes, but clearly related, clearly connected. Why would that be? Darwin thought about it. He realized that the living sloths were related to the extinct ones. They shared a common ancestor. Over time, some sloths had become smaller. They had adapted, they had evolved. The giant sloths had died out. But their smaller relatives survived. They changed, they evolved, they became the sloths we know today. This was a radical idea. Most people at the time thought species were created separately, that they never changed, that they were fixed, unchanging, perfect. But Darwin saw the connections. He saw the patterns. He saw that life was a family tree, all connected, all changing, all related, all evolving. He saw it in the finches. Different beaks, different islands, but all finches, all related. He saw it in the tortoises. Different shells, different islands, but all tortoises, all related. He saw it in the fossils. Extinct animals, living animals, but clearly related, clearly connected. Life was a family tree, a tree of evolution, a tree of change, a tree of connection. All life shared ancestors. All life was related. All life was evolving, always, constantly, beautifully. But he couldn't prove it with DNA. He couldn't show the genetic relationships. He couldn't read the code. He could only show the similarities, the patterns, the evidence, the observations. It was enough to change science. It was enough to revolutionize biology. But imagine if he had more. Imagine if he could have sequenced the DNA of those sloths, both the living ones and the fossils. Well, actually, DNA doesn't last that long in fossils. Most fossils are too old. The DNA has degraded, it's broken down. It's gone. But he could have sequenced living sloths. He could have compared their DNA to other animals. He could have seen the genetic relationships. He could have built a family tree based on genetics. He could have seen the exact connections, the proof written in code. He could have done the same with the finches. Sequenced their DNA, compared the codes, seen the genetic differences, understood how the beaks evolved, at the molecular level, in the code itself. It would have been incredible, revolutionary, undeniable proof. Darwin's method was simple but powerful. Observe, collect, compare, think. He filled notebooks with observations, page after page, volume after volume. He sketched what he saw. Beautiful drawings, detailed illustrations. He measured everything carefully. Lengths, widths, weights, everything. He asked questions. Why are these birds different? Why do these fossils look like living animals? What connects all these creatures? How did they get here? How did they change? What is the mechanism? How does evolution work? He didn't have fancy tools, no DNA sequencers, no electron microscopes, no computers, no genetic analysis, just his eyes, his hands, his mind, his notebook, his pencil, and his curiosity. That curiosity drove him. It made him look closer. It made him ask why. It made him see connections others missed. It made him discover truth. That's what great scientists do. They use whatever tools they have, but more importantly, they use their minds. They observe carefully. They think deeply. They ask good questions. They're curious. They're patient. They're dedicated. They don't give up. They keep searching. They keep asking. They keep thinking. Darwin did all of this. And he discovered one of science's greatest ideas: evolution by natural selection, the mechanism of change, the process of adaptation, the story of life. All without seeing DNA, all without reading the code, all with just observation, just thinking, just curiosity. But what is DNA, and how does sequencing work? How do we read the code of life? Let's explore that next. Let's dive into the secret instructions, the hidden code, the recipe book of life. DNA is like a recipe book. Every living thing has one. Every plant, every animal, every bacterium, every fungus, every living cell, they all have DNA. They all have the same basic code. The same 4 letters, the same instructions, the same mechanism. It's written in a special code, a code with just 4 letters, A, T, C, and G. That's it. Just 4 letters. But those 4 letters can spell out everything. Every trait, every characteristic, every protein, every function, everything that makes you, you. Everything that makes a finch, everything that makes a tortoise, a tortoise. These letters stand for chemicals: adenine, thymine, cytosine, guanine. They're called bases. They're the building blocks of DNA. They pair up in a special way. A always pairs with T, C always pairs with G. It's like a lock and key. A fits with T, C fits with G, always, never changes. These pairs form a ladder. The rungs of the ladder are the base pairs: A, T, C, G, A, T, C, G, over and over. Millions of times. Billions of times. The ladder is incredibly long. And it twists, it spirals. That's the famous double helix. It looks like a twisted ladder, a spiral staircase. A beautiful, elegant structure. Scientists discovered this structure in 1953. James Watson and Francis Crick figured it out. But they used data from Rosalind Franklin. She took X-ray images of DNA. Her images showed the spiral structure. They showed the double helix. Without her work, they might not have solved it. But together, they cracked the code. They revealed the structure. They opened the door to understanding DNA. Rosalind Franklin was a brilliant scientist. She was an expert in X-ray crystallography. She took incredibly detailed images of DNA. Her photo 51 showed the double helix clearly. It showed the spiral structure. It showed the pattern. It was the key to solving the puzzle. Watson and Crick saw her image. They understood what it meant. They built a model. They figured out the structure. They published their discovery. They won the Nobel Prize. But Franklin didn't. She died before the prize was awarded. Her contribution was crucial. Her work was essential. But she didn't get the recognition she deserved. Today we recognize her contribution. We understand how important her work was. We see how essential her images were. We appreciate her role in discovering DNA's structure. She was a great scientist. She made a crucial discovery. She helped open the door to understanding life itself. If you stretched out all the DNA in one of your cells, it would be about 2 meters long. 2 meters. That's longer than you are tall. But it's packed so tightly it fits inside a cell smaller than you can see, smaller than a grain of sand. How is that possible? The DNA is wrapped around proteins. Like thread around a spool, it's organized, it's compact, it's efficient. The DNA wraps around proteins called histones. These histones act like spools. The DNA winds around them. Then those spools wind around each other. Then those wind again. It's like a Russian nesting doll. Layers of organization. Levels of compaction. All fitting into a tiny space, all organized, all efficient, all beautiful. Every cell in your body has a copy of this recipe book, the same instructions, the same code. All 3 billion letters. In every single cell. Your skin cells have it, your muscle cells have it, your brain cells have it, your heart cells have it. Every cell. The same code. The same instructions, but different cells read different parts. A muscle cell reads the parts about making muscle proteins. It ignores the parts about making brain tissue. A brain cell reads the parts about making brain proteins. It ignores the parts about making muscle. It's all there, the whole recipe book. But each cell only reads what it needs. Only uses what's relevant. This is called gene expression. Some genes are turned on, some are turned off. Different cells have different genes active. That's why a muscle cell is different from a brain cell. They have the same DNA. But they read different parts, they make different proteins, they become different cell types. Scientists can now measure which genes are active in which cells. They can see what each cell is doing. They can understand how cells work. They can see how development happens. They can understand diseases. All from understanding gene expression. All from reading the code. All from DNA sequencing. It's like having a library with millions of books. Every cell has access to all the books. But each cell only reads the books it needs. A muscle cell reads books about muscles. A brain cell reads books about brains. They have the same library, but they read different books. That's how cells work. That's how life works. That's how DNA works. DNA tells cells how to make proteins. Proteins are the building blocks of life. They're like tiny machines. They do almost everything. They build structures. They carry messages. They fight diseases. They digest food. They transport oxygen. They contract muscles. They think thoughts. Everything your body does depends on proteins. And proteins depend on DNA. Your body makes thousands of different proteins. Each one has a specific job. Hemoglobin carries oxygen in your blood. Insulin controls your blood sugar. Antibodies fight infections. Enzymes digest your food. Collagen builds your skin. Every function, every process, every action, all depends on proteins. The code tells the cell which proteins to make, when to make them, how much to make. It's like a recipe. Step by step. Ingredient by ingredient. Instruction by instruction. The DNA code says, make this protein. At this time. In this amount. The cell follows the instructions. It makes the protein. It does its job. Life goes on. But here's the amazing part. The code is read in groups of 3 letters. These groups are called codons. Each codon codes for one amino acid. Amino acids are the building blocks of proteins. There are 20 different amino acids. They combine in different ways. They form different proteins. All from the same code. All from A, T, C, G. All from groups of 3 letters. It's elegant. It's efficient. It's beautiful. But here's the amazing part. DNA can change. Sometimes, when cells copy DNA, they make mistakes. The copying isn't perfect. A letter gets changed. A letter gets added. A letter gets deleted. These mistakes are called mutations. They're like typos in the recipe book. Mutations happen all the time, every time a cell divides. Every time DNA is copied, mistakes occur. Most are caught and fixed. But some slip through. They become permanent. They become part of the code. They become part of the organism. Most mutations don't matter. They're in parts of the DNA that don't code for anything important. They're silent. They don't change anything. They don't affect the organism. They're neutral. They just exist. They're part of the variation, part of the diversity. Some mutations are harmful. They break something. They cause problems. They might change a protein, make it not work, make it work wrong. They might lead to disease. They might cause the organism to die. These harmful mutations usually don't get passed down. The organism dies. The mutation dies with it. Natural selection removes them. But a few mutations are helpful. They improve something. They give an organism an advantage. Maybe a thicker beak that can crack harder seeds. Maybe a longer neck that can reach taller plants. Maybe resistance to a disease. Maybe better camouflage. Maybe faster running. These helpful mutations get passed down. The organism survives. It reproduces. The mutation spreads. Over time, it becomes common. That's evolution in action. That's natural selection at the genetic level. Scientists have seen this happen. In real time. In the lab. In nature. They've watched mutations occur. They've seen helpful ones spread. They've seen harmful ones disappear. They've watched evolution happen. In the code. In real time. It's beautiful. It's powerful. It's the mechanism of evolution. Right there. In the DNA. Darwin saw the results of these changes. He saw how species adapted, how beaks changed, how shells changed, how everything changed. But he couldn't see the DNA changes themselves. He couldn't read the code. He couldn't see the mutations. He could only see what the code produced, the different beaks, the different shells, the different shapes, the results, not the mechanism. But we can read DNA now. We can sequence it. Sequencing means reading the code. Letter by letter, A, T, C, G, one at a time. In order, from beginning to end. Scientists have machines that can do this. Incredible machines. Powerful computers. Sophisticated technology. They take a sample of DNA, a tiny sample, from a cell, from blood, from saliva, from a feather, from anything that has cells. Even from ancient bones, from fossils. If the DNA is preserved, they extract the DNA, they purify it, they prepare it, they get it ready for sequencing. Then they break it into pieces, small pieces, manageable pieces. They can't read the whole thing at once, it's too long, too complex, so they break it up. They create fragments. Each fragment is sequenced separately, each piece is read independently. Modern sequencers use different methods. Some use chemicals. Some use light. Some use electricity. But they all do the same thing. They read the code, letter by letter, base by base. They determine the order, A, T, C, G. Over and over. Millions of fragments, billions of letters, all being read, all being sequenced. Then computers put it all back together. Like reading a book one word at a time, then putting the whole story together. Like solving a puzzle, piece by piece, fragment by fragment. The computer finds overlaps. It matches fragments. It assembles the sequence, until you have the complete sequence, the entire code, all 3 billion letters, in order, from start to finish. It's like having a book torn into millions of pieces, then putting it back together, finding which pieces connect, which words go together, which sentences make sense. It's complex, it's challenging, but computers can do it. They can assemble genomes, they can reconstruct the code, they can read the recipe book. The first DNA was sequenced in 1977. It was a virus, a small virus called X174, only a few thousand letters. 386 letters to be exact. It took months, it was painstaking, it was slow, it required incredible patience, incredible skill, incredible dedication. But it worked. Scientists had read DNA for the first time, they had cracked the code, they had opened the recipe book, they had proven it was possible. The scientist who did it was Frederick Sanger. He developed a method, a way to read DNA, letter by letter, base by base. It was revolutionary, it was groundbreaking. It opened a new era, the era of DNA sequencing, the era of genetic analysis, the era of molecular biology. Sanger won the Nobel Prize for this work. Actually, he won 2 Nobel Prizes. One for sequencing proteins, another for sequencing DNA. He's one of only 4 people to win 2 Nobel Prizes. His method, called Sanger sequencing, was used for decades. It sequenced the first human genome. It opened the door to understanding genetics. It changed biology forever. Technology got better, faster, cheaper, more accurate. Scientists sequenced more things. Bacteria, yeast, worms, flies, mice. More and more organisms, bigger and bigger genomes, faster and faster, cheaper and cheaper. The technology improved. The methods got better. The machines got faster. The costs went down. New methods were developed. Better methods, faster methods, more accurate methods, automated sequences, computer analysis, high-throughput sequencing, next-generation sequencing. The field exploded, the technology advanced, the capabilities grew, the possibilities expanded. Now we can sequence a whole human genome in a day, one day, 3 billion letters, read, sequenced, understood. It costs hundreds of dollars instead of billions. We've come so far. So fast. In just a few decades. From months to days. From billions to hundreds. From impossible to routine. It's incredible. It's transformative. It's revolutionary. We can sequence anything now. Any organism. Any species. Any individual. We can compare, we can analyze, we can understand, we can discover. The code is readable. The recipe book is open. The instructions are clear. The mechanism is revealed. All because of sequencing. All because of technology. All because of science. Scientists have sequenced over 3,000 species of animals. Over 1,000 species of plants. Thousands of bacteria. Hundreds of viruses. The database grows every day. More sequences. More genomes. More understanding. More discoveries. We can sequence ancient DNA too. From fossils. From mummies. From preserved remains. We can see what extinct animals were like. We can understand how they're related. We can trace their evolution. We can read their code. Even after they're gone, even after millions of years, the code preserves their story, their history, their relationships. We can sequence disease-causing organisms. We can see how they evolve, how they become resistant, how they spread. We can track outbreaks, we can develop treatments, we can save lives. All from DNA sequencing. All from reading the code. All from understanding genetics. During the COVID-19 pandemic, scientists sequenced the virus constantly. They tracked mutations. They saw new variants emerge. They understood how it evolved. Over 15 million viral genomes were sequenced worldwide. Scientists shared the data. They tracked the spread. They understood the evolution. They developed vaccines based on the genetic code. They saved millions of lives, all because of DNA sequencing. All because we can read the code. All because we understand genetics. The speed was incredible. Scientists sequenced the first COVID-19 genome in January 2020. Just weeks after the outbreak began. They shared it immediately. Scientists around the world used it. They developed tests. They created vaccines. They saved lives. All from reading the code. All from DNA sequencing. All from understanding genetics. Cancer treatment has been transformed by DNA sequencing. Doctors can sequence tumor DNA. They can see the mutations that cause cancer. They can develop targeted treatments. They can personalize medicine. They can save lives. All from understanding the genetic code. All from DNA sequencing. Genetic testing is now routine. Parents can test their babies for genetic diseases. Doctors can test patients for disease risk. People can learn about their ancestry. All from DNA sequencing. All from reading the code. All from understanding genetics. It's amazing, it's powerful. It's life-changing. The Human Genome Project was a huge effort. Scientists from around the world worked together. The United States, the United Kingdom, France, Germany, Japan, China. Many countries, many scientists, over 20 institutions, thousands of researchers, all working toward one goal: to read all the DNA in a human cell, all 3 billion letters. The complete human recipe book. It started in 1990. Scientists estimated it would take 15 years. They thought it would cost billions. They thought it was almost impossible. But they tried anyway. They worked, they sequenced, they analyzed, they discovered. It took 13 years. From 1990 to 2003. 13 years of work. Thousands of scientists. Billions of dollars. Over $3 billion spent. But they did it. In 2003, they finished. They had read the entire human genome. All 3 billion letters. Sequenced, mapped, understood. It was a milestone. A triumph. A revolution. The project generated over 3 billion letters of sequence data, enough to fill 200 phone books, enough to stack books 200 feet high. All that information, all that code, all those instructions read, understood, available to scientists everywhere. It changed biology forever. What did they learn? They learned that humans have about 20,000 genes. That's fewer than scientists expected, much fewer. They thought we might have 100,000 genes, maybe more. They thought complex organisms needed more genes. But no, just 20,000. That's all it takes. 20,000 genes. To make a human, to make you, to make everything you are. Your brain, your heart, your muscles, your bones, everything, just 20,000 genes. It's amazing. Such complexity, such diversity, such capability. All from 20,000 genes. It shows how efficient evolution is, how elegant the code is, how powerful genes are, how much can be done with so little. It's beautiful. It's elegant. It's efficient. They learned that most of our DNA doesn't code for proteins. About 98% of it. It doesn't make proteins. So what does it do? That was a surprise. Scientists expected most DNA to code for proteins. But it doesn't. Most of it does other things. Some of it controls when genes turn on, when they're active, when they're silent, when they're expressed. It's like switches, like dimmers, like controls. They regulate the genes. They control the expression. They determine when proteins are made. They're essential. They're important. They're part of the mechanism. Some of it might be left over from evolution. ancient genes that don't work anymore, remnants of our evolutionary past, fossils in the code, traces of ancient genes, hints of our history, clues to our ancestry, written in the code, preserved in the DNA. Some of it we still don't understand. It's called junk DNA. But maybe it's not junk, maybe it does something important. Maybe it has functions we haven't discovered yet. Maybe it's regulatory. Maybe it's structural. Maybe it's something else entirely. We're still learning, we're still discovering, we're still understanding. But the most amazing thing, they learned that all humans are 99.9% identical. We're almost exactly the same. Out of 3 billion letters, only about 3 million are different. That's 0.1%, just 0.1% difference. Between you and me, between you and anyone else on Earth, between any two humans, we're almost identical. We're almost the same, we're almost clones. The differences are tiny, but those tiny differences matter. They determine our eye color, our height, our hair color, our skin color, our susceptibility to diseases, our talents, our abilities, our personalities, our preferences. Everything that makes you, you. Everything that makes each of us unique. All from 0.1% difference. It's incredible. Such small differences, such big results, such diversity. From such similarity, it's beautiful, it's amazing, it's evolution. And here's something Darwin would have loved. We can compare DNA between species. We can see how related they are. We can build family trees based on genetics. We can see evolution in the code itself. The changes, the mutations, the connections, the relationships, it's all there. Written in A, T, C, and G. The story of life, the family tree, the evolutionary history, all in the code. If Darwin had DNA sequencing, he could have done all of this in the 1830s. He could have sequenced the finches from different islands. He could have seen the exact genetic differences. He could have shown how the code changed, how mutations led to different beaks, how natural selection worked at the genetic level. He could have sequenced the tortoises. He could have seen how their DNA differed, how the code changed to produce different shell shapes. He could have sequenced the fossils. Well, actually, that's harder. DNA doesn't last that long in fossils. But he could have sequenced living relatives. He could have built genetic family trees. Imagine his book, On the Origin of Species, with DNA evidence. Imagine the proof, not just observations, not just patterns, but the actual genetic code. The instructions, the changes written in the molecules themselves. It would have been revolutionary. It would have silenced critics immediately. It would have changed biology forever. But he didn't have it. So he did something even more remarkable. He figured it out anyway. He used observation, he used logic. He used evidence. He built a theory that explained everything. And 150 years later, DNA sequencing proved he was right. Every discovery confirmed his ideas. The genetic code showed exactly what he predicted. Evolution was real, natural selection was real, and DNA was the mechanism. That's the power of good science. Good ideas stand the test of time. They get confirmed by new evidence, they get refined. But the core truth remains. Darwin saw the patterns, he understood the process. Even without seeing the code, he knew how it worked, and DNA sequencing showed he was absolutely right. Now let's see what DNA has revealed about life's family tree. The connections, the relationships, the story written in code. DNA sequencing has become Darwin's greatest proof. It shows us the family tree of all life, every species, every organism, all connected, all related. The code tells the story of how life evolved, how species split apart, how they changed over millions of years. How they adapted, how they survived, how they became what they are today. The code doesn't lie, it shows the relationships, it reveals the connections. It tells the evolutionary history. It's like a time machine, a genetic time machine. That lets us see into the past. That shows us how life changed. That reveals the family tree. All written in A, T, C, G. Take whales for example. Darwin thought whales came from land animals. He saw similarities in their bones, the structure of their flippers. The bones in their fins, they looked like modified legs, like arms and hands that had changed, that had adapted to water, that had become flippers. But many people didn't believe it. How could a land animal become a whale? It seemed impossible. A mammal, living on land, then moving to water, then becoming a whale. It didn't make sense. It seemed too big a change, too radical, too impossible. But DNA sequencing proved Darwin was right. Whales are related to hippos. Yes, hippos. Those big water-loving animals in Africa, those massive creatures that spend most of their time in rivers and lakes, those animals that look nothing like whales. But they're related, they're family, they share a common ancestor. Here's how we know. Scientists sequenced whale DNA. They read the code, all of it, every letter. They sequenced hippo DNA too. They read that code. Then they compared them, letter by letter. They looked for similarities. They looked for differences. They calculated how related they were. Geneticists found that whales and hippos share about 90% of their DNA. That's incredibly similar. More similar than whales are to other sea creatures. More similar than whales are to dolphins. More similar than whales are to seals. Whales and hippos share more DNA than whales share with any other sea mammal. That means they're more closely related. That means they share a more recent ancestor. Scientists published this discovery in 2009. They compared the genomes of whales, hippos, and other mammals. The genetic evidence was clear. The relationship was undeniable. While many people thought whales were related to other sea mammals, the DNA showed otherwise. Whales are more closely related to hippos. The code doesn't lie. The genetics prove it. The relationship is clear. The genetic evidence is clear. The code doesn't lie. Whales and hippos are cousins. Distant cousins, but cousins nonetheless. They share ancestors. They're part of the same family tree. The DNA proves it. The code confirms it. The relationship is undeniable. This means whales and hippos share a recent ancestor. Millions of years ago, there was an animal. It lived on land. It was probably a small, hoofed mammal. Some of its descendants stayed on land. They evolved. They became larger. They became hippos. Others moved to the water. They evolved. They became streamlined. They became whales. The fossil evidence confirms this. Paleontologists have found fossils of whale ancestors, animals that looked like land mammals, but they were adapting to water. Their legs were becoming shorter, their bodies were becoming longer, their tails were becoming flukes, their nostrils were moving to the top of their heads. They were becoming whales. One famous fossil is called Ambulocetus. It means walking whale. It lived about 50 million years ago. It had legs. It could walk on land. But it also had adaptations for water. Its legs were becoming flippers. Its body was becoming streamlined. It was in transition, half land, half water, a perfect intermediate. A missing link. Another fossil is called Basilosaurus. It lived about 40 million years ago. It was fully aquatic. It couldn't walk on land anymore. But it still had tiny hind legs, remnants of its land-dwelling past, evidence of its evolution, proof of its transformation. The fossils show the transition, step by step. generation by generation. From land to water, from legs to flippers, from land mammal to whale. It's a beautiful story. A story of adaptation. A story of evolution. And DNA shows the genetic relationship. It confirms the fossils. It proves the connection. It's perfect. Science confirming science. Evidence building on evidence. Truth revealed. Darwin would have loved this. He predicted it. He saw the patterns. He knew whales came from land. He wrote about it. He explained it. But he couldn't prove it with genetics. He couldn't show the DNA relationship. He could only show the bone similarities, the structural connections, the patterns. Now we can. DNA sequencing shows the exact relationship. The genetic code proves the connection. It shows that whales and hippos are cousins, distant cousins. But cousins nonetheless. They share ancestors. They're part of the same family tree. It's beautiful, it's elegant, it's science at its best. Observation, prediction, confirmation. Truth revealed. Here's another amazing discovery. Birds are dinosaurs. Yes, really. The chickens in your backyard? They're dinosaurs. The sparrows at your bird feeder? Dinosaurs. The eagles soaring overhead? Dinosaurs. The robins in the spring. The cardinals in the winter. The hummingbirds at your flowers. All of them. Every bird on Earth is a dinosaur. Every single one. How do we know? DNA and fossils together tell the story. Paleontologists have found fossils of dinosaurs with feathers. Beautiful fossils. Preserved in incredible detail. Over 50 different species of feathered dinosaurs have been discovered. Some looked almost exactly like birds. Small, feathered, lightweight. They could probably fly, or at least glide. They were the ancestors of modern birds. Archaeopteryx was one of the first feathered dinosaurs found. It lived about 150 million years ago. It had feathers, like a bird. But it also had teeth, and a long tail like a dinosaur. It was a perfect transition. A missing link, showing how dinosaurs became birds. The famous T-Rex, that massive predator, that king of the dinosaurs. Its closest living relatives are birds. Not lizards. Not crocodiles. Not any reptile. Birds. The DNA confirms it. The genetic code shows the relationship. T-Rex and chickens share a common ancestor. They're cousins. Distant cousins. But cousins nonetheless. Scientists have sequenced bird DNA. They've compared it to other animals. The genetic code shows birds are more closely related to dinosaurs than to any other living group. The relationship is clear. The code is definitive. Birds are dinosaurs. Living dinosaurs. The last survivors of a once dominant group. Think about that. When you see a chicken, you're looking at a dinosaur. A small one. A changed one. A domesticated one. But a dinosaur nonetheless. Evolution transformed them. Over millions of years they became smaller. They developed feathers. They learned to fly. They adapted to new environments. They diversified. They became the thousands of bird species we know today. But the DNA is still there. The code still shows the connection. The family tree still links them. The genetic relationship is clear. Birds are dinosaurs. Living dinosaurs. The last surviving dinosaurs. All the others went extinct. But birds survived. They evolved. They adapted. They thrived. And now they're everywhere. In every ecosystem. On every continent. Flying. Singing. Living. Dinosaurs. Darwin would have been amazed. He knew birds were special. He studied them carefully. He collected them. He observed them. He saw their diversity. He saw their adaptations. But he couldn't have imagined this connection. That birds are living dinosaurs. That the code would prove it. that genetics would reveal this incredible relationship. It's one of evolution's greatest stories, and DNA sequencing told it. The code revealed it. The genetic evidence proved it. Now what about us, humans? Where do we fit in the family tree? DNA sequencing has revealed our story too, our place in the tree of life. Our relationships, our history, our evolution. We share 98.8% of our DNA with chimpanzees. That's almost identical. Just 1.2% difference. But that tiny difference makes all the difference. It's what makes us human. It's what separates us from our closest relatives. It's what gives us our unique abilities, our bigger brains, our complex language, our tool use, our culture, our civilization. What does that percentage mean? It means that out of every 1,000 letters in our DNA code, 988 are the same as a chimp's. Only 12 are different, just 12 letters out of 1,000. That's how similar we are, that's how closely related we are, we're almost identical. But those 12 differences matter. They matter a lot. They determine our bigger brains, our ability to walk upright, our complex language, our tool use, our creativity, our curiosity. Everything that makes us human. Everything that makes us unique. All from 1.2% difference. It's incredible. Such a small change. Such a big result. That's evolution. Small changes. Big effects. Over time. Over generations. DNA also tells us when we split apart. Humans and chimps share a common ancestor. That ancestor lived about 6 to 7 million years ago. In Africa, it was probably a small ape. Some of its descendants stayed in the trees. They evolved. They became chimps. Others came down to the ground. They evolved. They became humans. We went our separate ways. We evolved differently. We adapted to different environments. We developed different abilities. But the code still shows the connection. We're family, distant cousins, but family nonetheless. We share ancestors. We're part of the same family tree, the tree of life. The tree of evolution. All connected, all related, all one family. DNA sequencing has also revealed human migration, how our ancestors spread across the world. The code tells the story. Different populations have slightly different DNA. By comparing these differences, geneticists can trace migration routes. They can see when humans left Africa, about 60,000 years ago, how they spread to Asia, to Europe, to the Americas, to Australia, to every corner of the globe. Genetic studies show that all humans outside Africa share common ancestors who left the continent around 60,000 years ago. The code preserves this history. It's written in our DNA, it's part of our genetic heritage. It tells the story of our journey, our migration, our spread across the planet. Geneticists have traced this migration using mitochondrial DNA, that's DNA passed down from mothers. They've also used Y chromosome DNA. That's DNA passed down from fathers. By studying these, scientists can follow the paths. They can see the roots. They can understand the journey, all from the code. All from genetics. All from DNA sequencing. The genetic evidence shows that humans left Africa in waves, not all at once, but in groups, over time. Some went to Asia. Some went to Europe. Some went to the Americas. The code tells this story. The genetics reveal this history. The DNA preserves this journey. They can see the genetic markers, the signatures, the traces left in the code. They can follow the paths, the routes, the journeys. They can see when populations split, when they mixed, when they adapted, when they evolved. Geneticists have mapped these migrations. They've traced the routes, they've followed the paths. All from DNA. All from the code. For example, genetic evidence shows that Native Americans came from Asia. They crossed a land bridge that connected Siberia to Alaska. This happened about 15,000 years ago. The DNA tells this story. The code preserves this history. It's like a genetic map. A map written in code. A map that shows our history, our journey, our family tree, our story. Darwin would have been fascinated. He knew humans were part of the animal kingdom. He knew we evolved, he wrote about it, he explained it. But he couldn't see the genetic evidence, he couldn't read the code, he couldn't trace the migrations. He couldn't see the relationships at the molecular level. Now we can. DNA sequencing shows it all. The connections, the relationships, the migrations, the adaptations. The story of life itself. Written in code. Revealed by science. Confirmed by genetics. It's beautiful. It's elegant. It's the story of us. The story of life. The story of evolution. Scientists use DNA to build evolutionary trees. They compare the code between species. They sequence the DNA, they read the code, they compare, letter by letter. They look for similarities. They look for differences. They calculate how related species are. The more similar the code, the more recently they shared an ancestor. The more different the code, the longer ago they split apart. It's like comparing recipes. If two recipes are almost identical, they probably came from the same cookbook. They share a source. They're related. If they're very different, they came from different sources. They're not closely related. The same is true for DNA. Similar code means close relationship. Different code means distant relationship. But it's more precise than recipes. DNA gives exact numbers, exact percentages, exact relationships. We can calculate when species split apart. We can estimate when common ancestors lived. We can build precise family trees. We can see the exact connections, the exact relationships, the exact history. DNA builds these trees automatically. The code doesn't lie. It shows the relationships. It reveals the family connections. It tells the story of evolution. It shows when species diverged. It shows how they're related. It shows the family tree, the tree of life, the evolutionary history. All written in code, all revealed by genetics. Darwin saw the patterns. He understood the process. He built family trees based on observations, based on similarities, based on patterns. But DNA shows the mechanism, the actual code, the instructions, the proof written in molecules. It confirms Darwin's trees, it refines them. It makes them precise, it proves them correct. It's beautiful, it's elegant, it's science confirming science, observation confirmed by genetics, patterns confirmed by code, theory confirmed by mechanism. Darwin's ideas were right. Evolution is real, natural selection is real, and DNA is the mechanism. The code that changes, the instructions that adapt, the recipe book that gets rewritten over time, the mechanism that drives evolution, the process that creates diversity, the force that shapes life. Scientists have now sequenced thousands of species. They've built comprehensive family trees. They've mapped the tree of life. They've seen how everything is connected, how all life shares ancestors, how evolution has shaped every species, how natural selection has driven change, all confirmed by DNA, all proven by genetics, all revealed by the code. The tree of life shows that all living things are related: bacteria, plants, animals, fungi, all connected, all part of one family tree, all evolved from common ancestors. all shaped by the same process. Evolution, natural selection, DNA, the code, the mechanism, the truth. Scientists have discovered surprising relationships. For example, fungi are more closely related to animals than to plants. That's right. Mushrooms are more like you than like trees. The DNA shows it, the code proves it, the relationship is clear. Genetic studies show that fungi and animals share a common ancestor. They split apart about 1 billion years ago, but they're still more closely related to each other than either is to plants. The code reveals this, the genetics prove it, the relationship is undeniable. Another surprise, whales are more closely related to hippos than to fish, even though they live in the ocean, even though they look like fish. Their DNA shows they're mammals, their code proves they came from land, their genetics reveal their history. Scientists have found that whales and hippos share about 90% of their DNA. That's incredibly similar, more similar than you might think. It means they share a recent ancestor, one that lived about 50 million years ago. The code tells this story. The genetics reveal this history. These discoveries are possible because of DNA sequencing. Because we can read the code. Because we can compare genomes. Because we can see the relationships. Because we can understand evolution at the molecular level. It's incredible, it's transformative, it's the power of genetics. Every day scientists make new discoveries. They sequence new species, they find new relationships, they understand evolution better. They see the tree of life more clearly. All because of DNA sequencing. All because we can read the code. All because we understand genetics. It's all there. In every cell, in every organism, the story of life, written in code. Revealed by science. Confirmed by genetics. Proved by DNA. The mechanism, the process, the truth. All in the code. All in A, T, C, G. All in the molecules. All in the instructions. The story of evolution. The family tree of life. The history of all living things. Written. Sequenced. Understood. Beautiful. Now let's imagine what might have happened if Darwin had DNA sequencing technology. Let's explore this what if, this alternate history of science. This journey into possibility. What if Darwin had DNA sequencing? What if he could have read the code of life in the 1830s? What if he could have seen the genetic differences, the mutations, the mechanism? How would that have changed everything? How would that have changed science? How would that have changed the world? Let's imagine, let's explore, let's see what might have been. It's a fascinating thought experiment. A journey into alternate history. A glimpse into a world where technology arrived earlier. Where understanding came faster. Where proof was immediate. First, the instant proof. Darwin spent 20 years gathering evidence. He studied barnacles for 8 years. He bred pigeons. He read everything. He corresponded with scientists. He built his case slowly. Carefully. Patiently. He wanted to be sure. He wanted to be right. He wanted to have enough evidence. He wanted to answer every question, every criticism, every doubt. But with DNA sequencing, he could have had proof immediately. He could have sequenced those Galapagos finches right on the ship. He could have seen the genetic differences, the exact changes in the code, the mutations, the variations, the mechanism, all right there, in the code, written in A, T, C, G. Imagine him on the Beagle. The ship is anchored off one of the Galapagos Islands. Darwin goes ashore. He collects a finch. He takes a small sample, a drop of blood, a feather, a tiny piece of tissue. He goes back to the ship. He has a DNA sequencer on board. A portable one. Modern technology in the 1830s. Impossible, maybe, but let's imagine. He sequences the finch's DNA, he reads the code, all of it, every letter. He writes it down, he labels it. Which island? Which finch? Which species? Then the ship sails to another island. He collects another finch, he sequences that DNA too. He compares them, letter by letter, he sees the differences, the mutations, the changes that led to different beaks. It's all there, in the code. Written in ATCG. The genetic differences. The mutations that make one beak thick and another thin. The code that determines the shape. The instructions that create the adaptation. He can see it, he can prove it, he can show it. The mechanism is clear. Evolution is real. Natural selection works. At the genetic level, in the code itself. He wouldn't have needed 20 years. He wouldn't have needed to study barnacles for 8 years. He wouldn't have needed to breed pigeons. He wouldn't have needed to gather more evidence. He would have had it. Right there, on the ship. In the code. The proof. The mechanism. The answer: he could have published his book in 1836, right after the voyage, as soon as he returned to England, with genetic proof, with the actual mechanism, with undeniable evidence, with DNA sequences, with genetic comparisons, with evolutionary trees built from code. It would have been revolutionary. It would have been immediate. It would have been undeniable. Critics couldn't have argued. They couldn't have said the mechanism was unknown. They couldn't have said evolution was just a theory. They couldn't have denied the evidence. The code doesn't lie. The genetic differences would have been clear. The mutations would have been obvious. The mechanism would have been proven. Evolution would have been accepted. Immediately. Completely. Universally. But that's not all. He could have discovered things that took us 150 years to find. Discoveries that revolutionized biology. Discoveries that changed our understanding. Discoveries that proved relationships. All in the 1830s. All with DNA sequencing. The whale-hippo connection? He could have found that in the 1830s. He could have sequenced whale DNA. He could have sequenced hippo DNA. He could have compared them. He could have seen they were related, more closely related than whales are to other sea creatures. He could have proven his prediction, that whales came from land animals, that they evolved from terrestrial mammals, that they adapted to water. The genetic code would have shown it. The relationship would have been clear. The proof would have been undeniable. The bird-dinosaur connection? He might have found that too. If he sequenced bird DNA and compared it to other animals, he might have seen the relationship. The genetic code would have shown it. Birds would have been more closely related to reptiles than to mammals. The family tree would have been clear. Birds are dinosaurs, living dinosaurs, the last survivors. Proven by genetics. In the 1830s. A discovery that took us until the 2000s to confirm. But he could have found it 170 years earlier. The human-chimpanzee relationship. He could have discovered that. He could have sequenced human DNA. He could have sequenced chimp DNA. He could have compared them. He could have seen we're 98.8% identical. Almost the same. Almost identical. He could have proven we're related. That we share a common ancestor. That evolution applies to humans too. That we're part of the animal kingdom. That we evolved. The genetic code would have shown it. The relationship would have been clear. The proof would have been undeniable. He could have discovered the genetic basis of inheritance. How traits are passed down. How characteristics are inherited. How genes work. How DNA codes for proteins. How mutations cause variation. how natural selection works at the genetic level, all in the 1830s. All with DNA sequencing, all with genetic analysis. This would have been revolutionary. Gregor Mendel discovered the basic laws of inheritance in the 1860s. But he didn't know about DNA, he didn't know about genes. He just saw patterns. He studied pea plants. He saw how traits were passed down. But he didn't know the mechanism. He didn't know about DNA, he didn't know about genes. If Darwin had DNA sequencing in the 1830s, he could have seen the mechanism. He could have understood how inheritance worked at the molecular level. He could have discovered genetics decades before Mendel. He could have seen how traits were encoded, how they were passed down, how they were inherited, all from the code, all from DNA, all from genetics. Mendel's work was important, it showed the patterns. But Darwin with DNA sequencing could have shown the mechanism. He could have seen the code. He could have understood how it worked. He could have revolutionized biology decades earlier. All because of one tool. All because of DNA sequencing. He could have built evolutionary trees based on genetics. By comparing DNA sequences, he could have seen how species are related. The code would have revealed the family tree of life. Genetic differences would have shown when species split apart. The molecular clock would have calculated when common ancestors lived. The entire tree of life could have been mapped. All from DNA. All from the code. All from genetics. Scientists today use something called the molecular clock. It's based on the idea that DNA mutations accumulate at a steady rate. By counting the differences between species, they can estimate when they split apart. It's like a genetic time machine. Darwin with DNA sequencing could have used this. He could have calculated evolutionary timelines. He could have dated the tree of life, all from the code, all from genetics. The molecular clock works because mutations happen at a roughly constant rate. Not exactly constant, but roughly. Over long periods of time, the rate averages out. Scientists can use this to calculate when species diverged, when they split apart, when they went their separate ways. It's like a genetic stopwatch, it measures evolutionary time. It tells us when species split apart. The molecular clock works because mutations happen at a roughly constant rate. Not exactly constant, but roughly. Over long periods of time, the rate averages out. Scientists can use this to calculate when species diverged, when they split apart, when they went their separate ways. For example, scientists have calculated that humans and chimps split about 6 to 7 million years ago. They did this by counting mutations, by comparing DNA. By using the molecular clock, the code tells the time. The mutations reveal the history. The genetics show the timeline. Darwin could have developed this concept in the 1830s. He could have calculated evolutionary timelines. He could have dated when species split apart. He could have built a timeline of evolution, all from the genetic code, all from DNA sequencing, all from understanding mutations. It would have been revolutionary. It would have been incredible. It would have changed everything. All of this in the 1830s. Instead of waiting until the 1970s, 1990s, or 2000s, biology would have advanced 150 years faster. We would understand genetics much earlier. We would understand evolution at the molecular level from the start. We would understand inheritance. We would understand variation. We would understand the mechanism. It would have changed everything. Science, medicine, agriculture, our entire understanding of life, all transformed, all revolutionized. All because of early DNA sequencing. But there's more. The Galapagos revolution. Darwin could have sequenced every species he found. Every finch, every tortoise, every plant, every insect, every lizard, every bird, every living thing. He could have built a complete genetic map, a comprehensive database, a full catalogue of the code. He could have seen exactly how they were related, how the code changed from island to island, how mutations led to new species, how evolution worked in real time. He could have sequenced finches from every island. He could have compared their DNA. He could have seen the genetic differences, the mutations that made beaks thick or thin, the code changes that created adaptations. He could have mapped the genetic variation. He could have seen how it matched the environmental differences. He could have proven natural selection at the genetic level. He could have sequenced tortoises from every island. He could have seen how their DNA differed, how the code changed to produce different shell shapes, how mutations created the saddle-shaped shells, how the code adapted to the environment, how genetics matched the observations, how the mechanism worked. He could have mapped genetic changes to environmental changes. This island has hard seeds. The finches here have genes for thick beaks. Specific mutations. Specific code changes. This island has soft fruit. The finches here have genes for thin beaks. Different mutations. Different code changes. This island has cactus flowers. The finches here have genes for specialized beaks. Unique mutations. Unique adaptations. He could have seen the connection, the code adapting to the environment. Natural selection at the genetic level. Mutations creating variation. Selection favoring helpful mutations. The code changing. The species evolving. The mechanism revealed. The process understood. The truth proven. He could have understood mutations immediately, what they are, how they happen, how they spread, how they create variation, how they drive evolution. He could have seen evolution in real time, not just the results but the process, the mechanism, the code changing, the instructions adapting, the species evolving. It would have been revolutionary, it would have been immediate, it would have been undeniable. He could have predicted evolutionary paths. If he understood the genetic code, he could have predicted how species might change, what mutations might be helpful, what adaptations might occur, what traits might evolve. He could have tested his predictions. He could have watched evolution happen, in the code, in real time, in the laboratory of nature. Imagine his notebooks, full of DNA sequences, thousands of sequences, millions of letters, full of genetic comparisons, species compared to species, island compared to island, full of evolutionary trees built from code, genetic family trees, molecular phylogenies, full of observations, full of data, full of proof. It would have been incredible. Science would have leaped forward, biology would have been transformed, genetics would have been born, molecular biology would have started, evolutionary genetics would have begun. All in the 1830s. All because one scientist had the right tool at the right time. All because DNA sequencing existed when Darwin needed it. All because technology and curiosity met. All because observation and tools combined. It would have been a revolution, a transformation, a new era of science. But he didn't have it. So he did something even more remarkable. He figured it out anyway. He used observation, he used logic, he used evidence. He built a theory that explained everything. And 150 years later, DNA sequencing proved he was right. Every discovery confirmed his ideas. The genetic code showed exactly what he predicted. That's the power of good science. Good ideas stand the test of time. They get confirmed by new evidence. They get refined. But the core truth remains. Darwin saw the patterns. He understood the process. Even without seeing the code, he knew how it worked. and DNA sequencing showed he was absolutely right. But what if he had had it? What if DNA sequencing existed in the 1830s? How would that have changed the world? Let's explore the ripple effects, the cascading changes, the transformations, the revolutions that would have followed. If Darwin had DNA sequencing, it wouldn't just change biology, it would change everything. Medicine, agriculture, conservation, education, technology, society. Our entire understanding of life. The ripple effects would be enormous. One technology, in the right hands, at the right time, changing the world, transforming civilization, revolutionizing human knowledge. Let's start with medicine. If Darwin had DNA sequencing in the 1830s, we would understand genetic diseases immediately. We would know why some diseases run in families. We would see the genetic mutations that cause them. We could develop treatments much earlier. Personalized medicine would arrive 150 years early. The medical revolution would begin in the 1800s, not the 2000s. Think about it. In the 1800s, doctors didn't understand why some diseases were inherited. They saw patterns, families with the same diseases, generations passing down the same conditions. But they didn't know the mechanism. They didn't understand how it worked. They couldn't explain it. They couldn't treat it. They couldn't prevent it. With DNA sequencing, they would. They could sequence a patient's DNA. They could find the mutation. They could see exactly what was wrong. They could understand the disease. At the molecular level, in the code itself, they could develop treatments. Targeted treatments. Specific treatments. Treatments that address the root cause, the genetic mutation, the code error. Diseases like hemophilia, sickle cell anemia, cystic fibrosis, Huntington's disease. All genetic. All caused by mutations. All could have been understood in the 1800s. With DNA sequencing, with genetic analysis, with molecular understanding. Take sickle cell anemia, for example. It's caused by a single mutation, one letter change in the DNA code, just one letter. But it changes the shape of red blood cells. It causes pain. It causes problems. Scientists didn't discover this until the 1950s. But with DNA sequencing in the 1830s, doctors could have found it immediately. They could have seen the mutation. They could have understood the disease. They could have developed treatments centuries earlier. Huntington's disease is another example. It's caused by a repeating sequence in the DNA. The same 3 letters repeated over and over. Too many times. This causes brain damage. It's fatal. Scientists discovered this in the 1990s. But with early DNA sequencing, it could have been found in the 1800s. The mutation would have been visible. The cause would have been clear. Treatments could have been developed. Lives could have been saved centuries earlier. Antibiotic resistance. We would understand it from the start. We would see how bacteria evolve, how their DNA changes, how mutations make them resistant, how natural selection favors resistant bacteria, how they spread, how they adapt. We could develop better antibiotics. We could prevent resistance. We could use antibiotics more wisely. We could save millions of lives. All because we understood genetics 150 years earlier. But that's not all. We would understand how diseases evolve, how viruses change, how they adapt, how they mutate, how they become more dangerous, how they spread. We could predict outbreaks. We could track mutations. We could develop vaccines faster. We could prevent pandemics. We could save countless lives. The medical revolution would be incredible, all starting from DNA sequencing in the 1830s. All because one scientist had the right tool at the right time. Now, agriculture. If we understood genetics early, we could breed better crops. We could see which genes make plants grow faster, Which genes make them resistant to disease? Which genes make them produce more food? Which genes make them drought resistant? Which genes make them nutritious? We could solve food problems centuries earlier. We could feed more people. We could prevent famines. We could improve nutrition. We could transform agriculture. The Green Revolution happened in the 1960s. New crop varieties, better yields, feeding billions of people, saving millions from starvation. It was a triumph, a success, a revolution. But if we had DNA sequencing in the 1830s, this could have happened much earlier. We could have developed better crops in the 1800s. We could have fed more people. We could have prevented famines. We could have improved lives centuries earlier. Imagine farmers in the 1800s. With DNA sequencing, they could sequence their crops. They could see which plants had the best genes. They could breed for specific traits. They could create better varieties, faster, more efficiently, more effectively. For example, they could identify genes that make wheat resistant to rust. Rust is a fungal disease that destroys wheat crops. It caused famines throughout history. Scientists didn't find rust-resistant genes until the 1900s. But with DNA sequencing in the 1800s, farmers could have found them immediately. They could have bred resistant wheat. They could have prevented crop failures. They could have fed more people. They could develop crops for specific climates. For dry regions, for wet regions, for cold regions, for hot regions. They could identify genes for drought tolerance, genes for cold resistance, genes for heat tolerance. They could breed crops adapted to local conditions. They could increase yields. They could improve nutrition, all starting in the 1800s. All because of early genetic understanding. The Irish potato famine happened in the 1840s. Over 1 million people died. It was caused by a disease that destroyed potato crops. With DNA sequencing, farmers could have identified disease-resistant genes. They could have bred resistant potatoes. They could have prevented the famine. They could have saved lives, all with early genetic knowledge. We would understand how plants adapt, how their DNA changes, how they evolve to fit their environments, how mutations help them survive, how natural selection shapes them. We could help them adapt faster. We could guide their evolution. We could create crops for specific needs, for specific soils, for specific conditions. Agriculture would be transformed. All because of early genetic understanding. All because of DNA sequencing in the 1830s. And conservation. If we understood genetics early, we could protect species better. We could see genetic diversity. We could understand how species adapt. We could develop better conservation strategies. We could save endangered species. We could protect biodiversity. We could preserve the tree of life. In the 1800s, species were going extinct. The passenger pigeon, the dodo, many others. But we didn't understand why. We didn't understand genetic diversity. We didn't understand how important it was. We didn't know how to protect it. We didn't know how to preserve it. With DNA sequencing we would. We could sequence endangered species. We could see their genetic diversity. We could understand how much variation they had. We could see if populations were too small, if they were inbreeding, if they were losing genetic diversity. For example, scientists have sequenced the DNA of cheetahs. They found that all cheetahs are almost identical. They have very little genetic diversity. Genetic studies show that cheetahs went through a population bottleneck. About 12,000 years ago, their numbers dropped dramatically. Only a few survived. All modern cheetahs are descended from those few. That's why they're so similar. That's why they have so little genetic diversity. This makes them vulnerable. They can't adapt well. They're at risk. Understanding this helps conservation efforts. It helps protect them. It helps save them. Conservationists use this genetic information. They make breeding decisions. They preserve diversity. They protect the species. All from understanding the code. All from DNA sequencing. We could make better decisions. We could protect the right habitats. We could breed species more effectively. We could preserve genetic diversity. We could save species from extinction. All from DNA sequencing. All from understanding genetics. All from reading the code. Scientists have used DNA to identify illegal wildlife trade. They can sequence samples. They can see what species they are. They can catch poachers. They can protect endangered animals. All from DNA sequencing. All from the code. All from genetics. We would understand how species are related. How their DNA connects them. How important genetic diversity is. For survival. For adaptation. For evolution. We could make better decisions. We could protect the right habitats. We could create better breeding programs. We could preserve the genetic heritage. The code of life. The recipe book of evolution. Conservation would be transformed. All starting from DNA sequencing in the 1830s. All because we understood genetics early. All because we could read the code. All because we could see the connections. All because we could protect what matters. The diversity. The variation. The code of life itself. But there's more. Education would change. If genetics was understood early, it would be taught in schools. Children would learn about DNA, about evolution, about the code of life, about how life works, about how species are related, about how we're all connected. Science education would be different, better, more complete, more accurate, more inspiring. Students would learn about the genetic code in the 1800s. They would understand how traits are inherited, how mutations create variation, how natural selection works, how evolution happens. They would see the mechanism. They would understand the process. They would appreciate the beauty, the elegance, the truth. Universities would teach genetics from the start. They would have genetics departments. They would have molecular biology programs. They would train geneticists. They would advance the field. They would make discoveries. They would push the boundaries. All starting in the 1800s. All because of early understanding. Other sciences would advance too. Chemistry would understand biological molecules better. They would study DNA structure. They would understand how molecules interact, how they form structures, how they function. Physics would study DNA. They would understand its physical properties. How it twists, how it folds, how it works. Mathematics would analyze genetic patterns. They would develop statistical methods. They would create models. They would understand probability. They would advance bioinformatics. All sciences would benefit. All because of early genetic understanding. Technology would spread. Once Darwin showed what DNA sequencing could do, others would want it too. Scientists around the world would use it. They would make discoveries. They would advance knowledge. They would push the boundaries. The scientific revolution would accelerate. Progress would be faster. Understanding would be deeper. Discovery would be constant. Scientists in Europe would use it. Scientists in America would use it. Scientists in Asia would use it. Scientists everywhere would use it. They would sequence everything. Plants, animals, bacteria, everything. They would compare. They would analyze. They would discover. They would understand. The global scientific community would be transformed. All connected. All advancing. All discovering. Together. But there would be challenges too. Ethical questions would arise early. Should we change DNA? Should we modify genes? Should we use genetics to improve humans? Should we create designer babies? Should we enhance intelligence? Should we eliminate diseases? These questions would come up in the 1800s. society would have to grapple with them earlier. Before we had the wisdom to handle them, before we understood the implications, before we had the ethical framework, there would be debates, there would be discussions, there would be conflicts, there would be challenges, but there would also be progress, there would be understanding, there would be wisdom, there would be growth. Society would evolve, ethics would evolve, understanding would evolve, all because of early genetic knowledge. Still, the benefits would be enormous. One technology, in the right hands, at the right time, changing medicine, changing agriculture, changing conservation, changing education, changing technology, changing society, changing everything. The ripple effects would be incredible. The world would be different, better, in many ways, more advanced, more understanding, more connected, more aware, more scientific, more curious, more inspired. But Darwin didn't have DNA sequencing, so we had to wait. We had to discover it ourselves. We had to build the technology. We had to learn how to use it. It took 150 years. But we got there, and now we're using it. We're making discoveries, we're advancing science, we're changing the world. The lesson: tools matter, technology matters, but so does the scientist using them. Darwin showed that. He didn't have DNA sequencing. But he had something else: curiosity, observation, patience, dedication. Those matter, too. Maybe more. Because good science comes from good thinking, not just good tools. Now let's explore the bigger picture. What really matters in science? What drives discovery? What makes great science? What is the essence of scientific understanding? The real story isn't just about DNA. It's not just about tools. It's not just about technology. It's about how scientists discover truth. How they use whatever tools they have. How they observe. How they think. How they ask questions. How they pursue knowledge. That's what matters. That's what makes great science. That's what drives discovery. Darwin's genius wasn't the tools. He didn't have DNA sequencing. He didn't have electron microscopes. He didn't have computers. He didn't have genetic analysis. He didn't have molecular biology. He had basic tools: a notebook, a pencil, a magnifying glass, a simple microscope, his eyes, his hands, his mind, and he made one of science's greatest discoveries. How? By observing carefully. By noticing patterns others missed. By asking good questions. By thinking deeply. By being patient. By dedicating his life to understanding. By not giving up. By persisting. By believing in the power of observation. That's what made him great. Not the tools, the thinking, the curiosity, the dedication. He spent years studying barnacles, 8 years, from 1846 to 1854. Why? To understand variation, to see how species could change, to see the differences, to see the patterns, to understand evolution. He collected thousands of specimens, over 10,000 specimens, from all over the world. He studied them under a microscope, he compared them, he saw the variations, he understood how they evolved. Darwin wrote 4 books about barnacles, 4 entire books. Just about barnacles. Over 1,000 pages total. He became the world's leading expert on them. He saw how they varied, how they adapted, how they evolved. This work helped him understand evolution. It showed him how species could change. It proved that variation existed. It supported his theory. It gave him confidence. It prepared him to publish. He bred pigeons. Why? To see how selection worked, to see how traits were passed down, to see how artificial selection could change species. to understand inheritance, to see the mechanism, to prove that selection worked. Darwin joined pigeon breeding clubs in London. He talked to breeders. He learned their methods. He saw how they selected for specific traits, how they created new varieties, how they changed species through selection. He bred pigeons himself. He saw how traits were inherited, how characteristics passed from parents to offspring, how selection could change a species. If artificial selection worked, why wouldn't natural selection work too? The mechanism was the same. Only the selector was different. In artificial selection, humans choose. In natural selection, nature chooses. But the process is the same. The results are the same. Species change. They evolve. They adapt. He read everything. Books, papers, letters. Why? To learn, to understand, to build his knowledge, to see what others had observed, to connect ideas, to build on previous work, to understand the bigger picture. He was dedicated, he was patient, he was curious, he was persistent, he was a true scientist. That's what great scientists do. They use whatever tools they have. But more importantly, they use their minds. They observe, they think, they ask questions, they're curious, they're patient, they're dedicated. They don't give up, they keep searching, they keep asking, they keep thinking. That's what matters, that's what drives discovery, that's what makes great science. Tools help. DNA sequencing is amazing. It reveals things we couldn't see before. It proves relationships. It shows evolution in action. It reads the code of life. It's powerful, it's transformative, it's revolutionary, but tools are just helpers. They're not the science. The science is in the thinking, in the questions, in the curiosity, in the observation, in the understanding. Modern scientists use DNA sequencing every day. They make incredible discoveries. They sequence genomes. They compare species. They build evolutionary trees. They understand diseases. They develop treatments. They advance knowledge. It's amazing. It's powerful. It's transformative. But they still need to think. They still need to ask questions. They still need to be curious. They still need to observe. They still need to understand. Tools don't replace thinking. They enhance it. What tools will future scientists have? We don't know, but we can imagine. Maybe they'll sequence DNA instantly. Maybe they'll read genomes in seconds. Maybe they'll understand the code completely. Maybe they'll predict evolution. Maybe they'll design new life forms. The possibilities are endless. The future is exciting. But we know they'll need to think. They'll need to observe. They'll need to ask questions. They'll need to be curious. They'll need to understand. That never changes. That's what science is. Thinking, questioning, discovering, understanding. That's the essence. That's what matters. Tools will get better. Technology will advance. Capabilities will grow. But the core will remain. Observation, thinking, curiosity, questions, understanding. That's what makes science. That's what drives discovery. That's what changes the world. That's what Darwin showed us. That's what great scientists demonstrate. That's what matters most. The best science combines old methods with new tools. Darwin's methods still work. Observation, comparison, careful thinking, patient study. Dedicated research. Those never go out of style. They're timeless. They're essential. But modern tools enhance them. DNA sequencing shows what observation suggests. It proves what thinking predicts. It reveals what curiosity questions. It confirms what understanding proposes. That's the combination. Old wisdom, new tools, together. That's how science advances. Each generation builds on the last. Each generation adds new tools. Each generation makes new discoveries. But the core remains. observation, thinking, curiosity, questions, understanding. That's what science is, that's what it always has been, that's what it always will be. Darwin shows us this. He didn't have modern tools, but he had the essentials. He observed, he thought, he asked questions, he was curious, he was patient, he was dedicated, and he changed the world. He discovered one of science's greatest ideas. He explained how life works. He showed that evolution is real. Then, 150 years later, modern tools proved he was right. DNA sequencing confirmed his ideas. It showed the mechanism, it revealed the code. It proved the relationships. It confirmed the family tree. It validated everything he had proposed. The tools confirmed the thinking. The technology proved the ideas. The code validated the observations. But the ideas came first. The thinking came first. The observation came first. The understanding came first. The tools just confirmed it. That's the lesson. Tools are helpers. They're powerful, they're amazing, but thinking is what matters. Curiosity is what matters. Observation is what matters. Understanding is what matters. That's what makes great science. That's what drives discovery. That's what changes the world. So what can we learn? Observe carefully. Think deeply. Ask good questions. Be curious. Be patient. Be dedicated. Use whatever tools you have. But remember, the tools are just helpers. The real science is in your mind, in your curiosity, in your thinking, in your understanding. That's where discovery happens. That's where truth is found. That's where science lives. That's what Darwin teaches us. That's what great scientists show us. Tools matter, technology matters, but thinking matters more. Observation matters more. Curiosity matters more. Understanding matters more. That's what drives discovery. That's what advances science. That's what changes the world. That's what makes great science. That's the essence. That's the truth. And now, as we come to the end of our journey, let's remember what we've explored. Let's reflect on what we've learned. Let's appreciate the story we've told. We've traveled through time. We've visited Darwin on the HMS Beagle. We've stood on that deck with him. We've felt the ocean breeze. We've seen the unknown lands ahead. We've experienced his curiosity, his wonder, his dedication. We've seen what he discovered with just his eyes and his mind, the finches with different beaks, the tortoises with different shells, the fossils that looked like living animals, the patterns, the connections, the truth, all discovered through observation, through thinking, through curiosity. We've learned how DNA sequencing works, how it reads the code of life, how it reveals the instructions, how it shows the mechanism, how it proves the relationships, how it tells the story of evolution, the story of life, the story of us. We've seen how DNA has confirmed Darwin's ideas, how it proved he was right, how it showed the mechanism he could only imagine, how it validated his observations, how it confirmed his theory, how it revealed the truth he had discovered. We've imagined what might have been if he had this incredible tool, the instant proof, the immediate acceptance, the early discoveries, the accelerated progress, the transformed world, the different history, the alternate reality. Darwin discovered evolution through careful observation. He saw patterns, he connected dots, he built a theory that explained everything. He did this without DNA sequencing, without modern tools, without genetic analysis, with just his curiosity, his patience, his dedication, his eyes, his mind, his notebook, his pencil. DNA sequencing reads the code of life. It shows us the instructions, the mechanism, the proof. It confirms Darwin's ideas. It shows evolution in action. It reveals the family tree of all life. It's amazing. It's powerful. It's transformative. It's changed biology forever. It's changed medicine. It's changed agriculture. It's changed our understanding of life itself. With DNA, Darwin could have revolutionized science immediately. He could have had proof. He could have silenced critics. He could have discovered things that took us 150 years to find. Biology would have advanced faster. Medicine would have advanced faster. Agriculture would have advanced faster. Conservation would have advanced faster. Everything would have been different. The world would have been transformed centuries earlier. But tools help. They don't replace thinking. Darwin showed that. He didn't have DNA sequencing, but he had something more important: curiosity, observation, patience, dedication, understanding. Those matter. Those drive discovery. Those make great science. Those change the world. Science builds on itself. Each generation adds new tools. Each generation makes new discoveries. Each generation advances knowledge. But the core remains: observation, thinking, curiosity, questions, understanding. That's what science is. That's what drives it forward. That's what makes it great. Darwin shows us that observation and thinking are powerful. They can discover truth, even without the best tools. They can reveal secrets, even without modern technology. They can change the world, even with just a notebook and a pencil. Modern tools let us see what Darwin could only imagine. They confirm his ideas. They reveal new truths. They advance science, but they don't replace thinking. They enhance it. They support it. They confirm it. Keep asking what if. That's how science advances. What if we could see DNA? What if we could read the code? What if we could understand evolution at the molecular level? What if we could see the mechanism? What if we could prove the relationships? These questions drive discovery. They push science forward. They change the world. They reveal truth. Thank you for exploring this journey with me. Through biology and history, through observation and technology, through the past and the present, through what was and what might have been, through time and space, through curiosity and understanding, through science and wonder. If you enjoyed this calm exploration of science and history, there are more stories waiting, more mysteries to explore, more questions to ask, more science to discover, more journeys to take, more truths to find, one sleepy soul at a time. Sweet dreams, and keep asking questions about the amazing world around us. The code of life is written in every cell, in every organism, in every living thing. The story of evolution is written in every species, in every adaptation, in every connection. And the power of curiosity is written in every great scientist, in every discovery, in every truth, including Darwin, including you, including all of us. The code is there, the story is there, the truth is there, waiting to be read, waiting to be understood, waiting to be discovered, by curious minds, by patient observers, by dedicated thinkers, by scientists, by you. Good night, and may your dreams be filled with wonder and curiosity. May you see the patterns, may you ask the questions. May you discover the truth, one observation at a time, one question at a time, one discovery at a time. The code of life is written, in every cell, in every organism, in every living thing. The story of evolution is told, in every species, in every adaptation, in every connection. The power of curiosity is eternal, in every scientist, in every discovery, in every truth. Darwin showed us that observation matters, that thinking matters, that curiosity matters. DNA sequencing showed us the mechanism, the code, the truth, Together they reveal the story of life, the story of evolution, the story of us. And science continues, always, forever, beautifully. Each generation builds on the last. Each discovery leads to the next. Each question opens new doors. Each observation reveals new truths. Science is a journey, a never-ending journey, of discovery, of understanding, of wonder. So keep observing, keep thinking, keep asking questions, keep being curious. That's what Darwin did. That's what great scientists do. That's what drives discovery. That's what changes the world. The code is there, waiting to be read. The story is there, waiting to be told. The truth is there, waiting to be discovered, by you, by scientists, by curious minds everywhere. Good night, and may your dreams be filled with wonder. May you see the patterns, may you ask the questions, may you discover the truth, one observation at a time, one question at a time, one discovery at a time, one sleepy soul at a time. Good night.